This is the second part of your genetic engineering notes, the applications of genetic engineering. So we're going to talk about 5 applications today. And let me get my cursor, the pen. So we'll talk about DNA fingerprinting, genetic engineering, making transgenic organisms, which we talked about last video, cloning, the Human Genome Project, and gene therapy. You might or might not know that what the sheep is. This is the first cloned sheep. This is Dolly that I told you that we'd talk about. And then we can talk about genetic engineered foods. These are just kind of labels that you'll see all the time. This is not necessarily saying that GE foods are bad. And in fact, most GE foods are just fine. Some have a lot of environmental impacts that are worth talking about. But again, this is not saying that Mr. Troutman does not support GE foods or anything like that. So in person, we would actually have a little debate about GE foods, but I'm not sure we'll be able to get to it this year. So if you do have questions for me, by all means, ask me in class and we can talk about them. All right, so every individual has unique DNA, which means if you take a sample of that DNA, cut it with restriction enzymes, separate it out using gel electrophoresis, and then look at those banding patterns, that banding pattern, just like a barcode, is unique to any organism, any individual, right? So this was actually a real case between two people. This is one of the first times we used this, and like mainstream kind of public awareness got caught up in it. This is the output of a gel electrophoresis that was run between two suspects, Chris and Randall, and then they had the crime scene sample, which was a semen sample, and it's really just as simple as matching the bars up. In this case, the bars matched Randall, and that was used in court as forensic evidence linking Randall to this sample of DNA. It didn't mean that Randall committed the crime. It increases the likelihood of it. It just means that his DNA is the same as the sample that they obtained. Then you would have to go about it in court and kind of prove things and disprove things and stuff like that. So that's how you would read the output of a DNA fingerprint. And this kind of banding pattern is called a DNA fingerprint. It's not like an actual fingerprint that you put ink on your finger or anything. This is called a DNA fingerprint. It is the output of gel electrophoresis. All right. Making a transgenic organism is actually more technical than this now. So what we're talking about here is a technique that was used up until the last decade. And I'll talk to you about what's replaced that in class. But let's go ahead and talk about this because it is still part of the curriculum. Again, a transgenic organism just means that it has DNA from another species in it. And one way to go about this is to make something called recombinant DNA. So recombinant DNA is a piece of DNA that has the genes from a different species in it. And we usually do this with bacteria. And that means we have to talk about plasmids. So here's a bacterial cell. Remember that bacteria are prokaryotic. They don't have a nucleus, so their DNA floats around the cytoplasm. So that's what this orangey stuff is. But bacteria also have a circular chromosome. They have a circular chromosome that this arrow is pointing to. That's a plasmid. That's what a plasmid is. It's a circular chromosome found in bacteria. This is what we use to make recombinant DNA. This is the piece of bacterial DNA that we're going to cut and insert other genes into. So here's an example of how we can make human growth hormone, HGH, using bacterial plasmids. So let's just look at the top first. Here's a human cell. Here's the nucleus. And once we've sequenced it, which we have, and identified the gene for human growth hormone, We use a restriction enzyme, EcoR1, we saw it last video, to cut the DNA on either side of the gene. So this purple section right here is the gene. So we cut on both ends. We have a piece of DNA here, and we take this DNA and we just set it aside. We just put it off to the side and chill out for a second. Let's look at the bacterial cell now. We take the plasmid of the bacterial cell, and using the exact same enzyme, and this is important because if we used a different enzyme, it would cut in a different spot. So the same restriction enzyme cuts the plasmid, opens the plasmid up, and produces these sticky ends. And because we cut both the human gene and the bacterial DNA with the same restriction enzyme, these sticky ends match, right? So these sticky ends on the human DNA and the bacterial DNA will match because they were cut with the same restriction enzyme. So you can just snap this together. Then you just put both of these in a solution, and because these are exposed bases, they will want to repair back up with their complements. So this can snap right back in place. The human gene is now inserted into your plasmid, and we have made a transgenic piece of DNA. So this is technically called recombinant. Recombinant, we've recombined the DNA. So we have a bacterial circular plasmid, which is just it's a chromosome, a circular chromosome with a human gene in the middle of it. Now, why would we want to do that? Well, we would do that because bacteria divide very, very quickly. They divide, once every 20 minutes. So in just an hour or so, you can have huge, multi-million celled colonies of bacteria. And every time bacteria divide, every time they do mitosis or binary fission, they also copy their plasmid, which means they also copy the human gene. So we let the bacteria mass produce this gene for us. And every time they transcribe and translate the genes on this plasmid, guess what? They'll also transcribe and translate the gene for human growth hormone. So bacteria mass produce human growth hormone for us. We take advantage of the fact that they are little factories that pump out proteins after proteins after proteins really, really quickly en masse. And it's a clever technique. All right, so that's how you make transgenic bacteria. And you can do this, again, you can do this with human growth hormone, you can do it with insulin, you can do it with blood clotting factors, and it's because bacteria grow and reproduce quickly, right? You can also do this with, yep, I was right, it was lightning bugs. Tobacco plants with GFP, green fluorescent protein from lightning bugs or jellyfish. You can make them glow. Feel free to Google glowing rabbits as well. We've made transgenic pigs. We've made transgenic sheep. They can, we can fortify milk by adding human proteins or by, again, changing the genes that when they're transcribed and translated make the proteins in the pigs and the sheep and then in the milk that they make. We can do this with corn and soybeans. These are actually one of the two most common genetically modified foods. Again, not all genetically modified foods are bad. By and large, genetically modified foods are actually just fine. There are a couple, like genetically modified Roundup Ready soybeans, that have harmful effects on the environment because of how they deal with pesticides. But in terms of human health, GMOs are really not the big baddie that a lot of people think they are. And you're going to have to go GMO by GMO because each GMO is slightly different. The effect of that gene is slightly different, but they are not nearly as bad as a lot of people think they are. Another example here is golden rice. Golden rice is transgenic rice with genes to help make additional vitamin A. This is not something we use in the US because we don't have a systemic vitamin A deficiency, but it can be used to help fortify the diets of people who do lack vitamin A. And it just gives the rice a kind of a goldeny color. That's why they call it golden rice. Oh, and these page numbers, by the way, they refer to the old textbook, so I actually have to update these page numbers. I can just give you articles in class that talk about GM foods, and actually I will do that, and we can refer to that and look up those examples. So for cloning, remember the first individual clone that we talked about in the last video was a sheep named Dolly. This is well before you guys were born in 1997. Didn't live too long, but lived long enough to be considered a successful clone. So I'm going to walk you through the steps. Again, you can kind of pause and look over the steps here, but it makes more sense to see a picture and diagram of it. So here's how they did it. There are actually 4 sheep. We're going to number these sheep. So this is sheep number 1. This is sheep number 2. This is sheep number 3, and this is sheep number 4. This is the actual clone. This is, in this case, this was Dolly. And we'll go through why there are different sheep, but sheep 4 is a clone of sheep 1. And we're going to walk through that now. So what we're going to do is take a cell from the sheep's udder, and we take it from the udder just because it's easily accessible, and it's, more importantly, diploid. It has all of the chromosomes necessary to build a body, right? It's not a haploid gamete cell. It's a diploid somatic cell. And we don't really care about the cell, but we do care about the nucleus, because that's where the nucleus, or that's where the DNA is. So we actually, well, sometimes you can just take the nucleus out, but really it's easier, and in this case they just kept the entire cell. But the point is, the diploid nucleus is there. Meanwhile, go to sheep number 2. Sheep number 2, we're going to take an egg cell, because during fertilization, the cell that gets fertilized is an egg cell. It's not a skin cell or anything like that. And we need to use this cell for fertilization. So we're taking the egg cell, but we don't want the DNA from sheep 2. We don't. So we take the nucleus out. This is a very, very delicate process that fails more than it succeeds. But when it does succeed, it's really cool. So when you take the nucleus out without killing the cell, you basically have a cell with cytoplasm and ribosomes and mitochondria and all that good stuff, but just no nucleus and no DNA. So we take the DNA from sheep 1 and we stick it into this egg cell. Right? So what we have is a cell with the DNA from sheep 1, but the cytoplasm from sheep 2. The cell is technically an egg cell, but it has the DNA of a diploid somatic cell. So what you're going to say is, well, Mr. Troughton, there's no need to fertilize this egg because the nucleus isn't haploid. It's a diploid nucleus, like you were just saying. And that's exactly right. This is a diploid nucleus, and we don't need a sperm to fertilize. Remember, this is a clone. There's no sperm or male sheep involved. So what we have to do is, when we insert the nucleus into this egg cell, we use an electric shock. And that does 2 things. The first thing it does is it helps the cell uptake the nucleus, first of all. But the second thing it does is it tricks the cell into thinking it got fertilized. It tricks the cell into thinking it got fertilized. So this cell will start to divide because it's turning into an embryo, right? So once the embryo is, I'm not sure what the exact cell count is, but probably around 16 to 32 cells, it is put inside the uterus of a foster mother, which is sheep number 3. And then when it's born, it's a clone of sheep number 1, because that's who gave the DNA. So let's go over this one more time. You can pause, you can slow the video down, but here we go. 4 sheep. Sheep number 1 gives the nucleus. Sheep number 2 gives the egg cell. Because sheep number 1 is the sheep we're cloning, we need to put this nucleus into this sheep's egg cell. So we take the nucleus out of the egg cell from sheep 2, discard it, we don't need that nucleus, and we insert the nucleus from sheep 1. Now we have a cell, an egg cell, where the cell itself is from sheep 2, but the DNA, the nucleus, is from sheep 1. Pretty cool. This divides into an embryo. That gets put into a carrier, a foster mother. And then when she gives birth, this lamb, this clone, is of sheep 1, because that's the sheep that gave the DNA. All right, I'm going to let you reread or read this on your own. I'm not going to talk too much about this. The Human Genome Project was completed about 2 decades ago at this point. 2 billion is actually, or 2 to 3 billion is pretty cheap, but this has allowed us to identify the genes of a whole host of diseases that we can now use genetic engineering to treat. So a major, both scientific but also medical breakthrough for having sequenced the human genome. And we've sequenced the genomes for a lot of other species as well. Dogs, cats, really any bird, frogs, fish, whatever you want, bacteria. There are databases called BLAST, B-A-L-S-T, that have all of this public information on them about these genomes. Super interesting. Just to give you some ideas, in humans we have about 35,000 genes, 3 billion base pairs, right? So this, yeah, it's all free. It's available to everyone. By all means, we can try to do this in class and do some research on it. I'm not sure if we can or if we'll have time. It's not part of the main curriculum. It's kind of an extra thing. But by all means, you know, look around, see if you can find some stuff out there. Make sure it's reliable information. And I can give you a reading on the Human Genome Project too that we can do. All right, the last thing, gene therapy. So taking everything we've talked about, what we are starting to do, and this is especially true for cancer research, is identify defective or broken or mutated copies of genes and replacing them with a good or working, functional, normal copy. We could not do this without genetic engineering and having sequenced the human genome, right? So we can use viruses in addition to bacteria to deliver genes. I will talk to you guys later about the technology called CRISPR. It's another way to do this that achieves the same outcome of putting in new genes and taking out bad ones. But we can do this with previously incurable diseases, like things like cystic fibrosis, hemophilia, autoimmune disorders, previously terminally ill death sentences are now being fixed by science and technology and gene therapy. So incredibly interesting stuff. Don't worry about this back page too much. You're free to look at it. I'm not going to focus on it too much this year just because of the circumstances and the time that we have. A lot of these techniques do involve stem cells because stem cells can divide and become any kind of cell you need them to be. So stem cells are incredibly valuable in scientific research. Adults have them. The reason you can make skin over and over, I mean, every day you slough off thousands and millions of skin cells. The reason why your skin doesn't disappear over the course of your life is because you have stem cells that keep on differentiating over and over again. Same thing with your blood cells. Liver cells can do this to some extent. They kind of behave like stem cells. They can regenerate and fix damaged tissue like a stem cell could. So stem cells are incredibly powerful and incredibly useful for curing diseases that we never would have imagined not 5 decades ago could have been cured. So you can look over this. We'll talk about CRISPR later on. I hope this helped, and I will see you in class.